Hello students, now let us study about the sexual reproduction in marcantia. What is sexual reproduction? It is a type of reproduction which involves the formation of gamete. The gametes are of two types, that is the male gamete and the female gamete. Okay, here the gametes are haploid in nature and these gametes undergo fusion to form diploid zygote through the process called as fertilization. This is a process involved in the sexual reproduction, right? Even in marcantia, marcantia, okay, are the gametophytic plant bodies which produces the gamete. Even in marcantia, they produces the gamete. The gametes are produced on a vegetative structure called as gametophores. The gametes are produced on the vegetative structure called as gametophores. These structures, what you see in the in the slide, okay, these structures are called as the gametophores. What is gametophores? The gametophores are the structures, long stalked structures, which is produced onto the dorsal surface of the marcantia, okay, which produces the gametes, are called as gametophores. What is gametophores? Gametophores are the gamete producing organs. Okay, here they are long stalked, they are very long. Okay, so the length of the gametophore might vary from 1 to 3 centimeters. They are attached onto the dorsal surface of the marcantia. As we studied on the morphology of the marcantia, marcantia is dorsiventrally flat. On the dorsal surface you can find the midribs. Okay, on the midribs they develop the stalked structure, and this stalked structure is called as the gametophores. The male gamete producing gametophore is different from the female gamete producing gametophores. The male gamete producing gametophores are called as the antheridiophores, okay, antheridiophore, and the female gamete producing gametophores is called as archegoniophores. Antheridiophores are developed on different plant body and the archegoniophores are developed on different plant bodies. Since antheridia, male gamete is produced on the different plant body and female gametes are produced on the different plant body, they are called as dioecious in nature. So marcantias are usually dioecious in nature because male gamete producing plant body is different from the female gamete producing plant bodies, and even the structure also is differs. And now coming on to the male gamete, the male gamete are produced on the antheridiophores, and the male gamete is smaller in size. The male gamete is smaller in size and they have the flagella. Since they have the flagella, they are motile. So how are the male gametes? They are small, flagellated and motile in nature. When you come to the female gamete, the female gametes are larger. They do not have flagella and they are non-motile. Here, small flagellated motile male gamete, which is normally considered as a sperm, move towards the bigger non-motile female gamete for the fertilization process, where male gamete fuses with the female gamete, they undergo fertilization. And this type of fertilization is called as oogamous type of fertilization. What is oogamous type of fertilization? Oogamous type of fertilization is a type of sexual fertilization, or it is a type of sexual reproduction where the male gamete is small, flagellated and motile, which moves towards the female gamete. They undergo fusion and okay, female gamete which is non-motile, they undergo fusion, they forms the fertilization or diploid zygote. That is what is the process. Now let us study, okay, so that is the process. What is sexual reproduction? How is the sexual reproduction in marcantia occurs? The sexual reproduction in marcantia is oogamous type. They involves the formation of gametes, that is antheridia and archegonia. Okay, they reproduce. The archegonia and antheridia are produced on a specialized structure called as gametophores. The gametophores are of two types, that is antheridiophores and archegoniophores. Antheridiophores are the gametophore which produces the male gamete, and the archegoniophores is the gametophores which produces the female gamete. Okay, so this is about the sexual reproduction. Now, if at all the sexual reproduction has to occur, The gametes has to be produced. We'll now see how the gametes are produced. First, we'll focus on the production on male gamete, that is antheridia. Before studying the antheridia, we have to study about the antheridiophore. As we know, antheridiophores are the gametophores which produces the male gamete, okay, in a specialized structure called as antheridium. So how is this antheridiophores and where it is present? As we know that marcantia is dorsiventrally flat on the dorsal surface in the midrib region, and it develops a long stalked structure, okay, which is called as antheridiophores. The antheridiophores are attached. One end of the antheridiophore is attached to the gametophyte, vegetative part of the gametophyte, and the other end of the gametophore is disc-like structure, and it has a long stalk. One end of the stalk is attached to the marcantius vegetative thallus, and the other end develops a disc-like structure. The disc is lobed, okay, they are lobed into 4 to 8 lobes. On the lobed structure, they develop a specialized organ called as antheridium, and in the antheridium the antheridiophores are developed. So this is what is given here. So what is antheridiophore? Antheridiophores are the gametophore which produces the male gamete, that is antheridium, antheridia. How are they? They are the modified vegetative thallus. They are long stalked. The stalk is around 1 to 3 centimeters in its length, and the other end of the stalk is one end of the stalk is attached to the vegetative gametophyte of the marcantia, and the other end of the stalk is disc shaped. The disc is lobed. The lobes are around 4 to 8 lobes, okay, on which the antheridium is produced. Antheridia is developed into antheridia. Antheridium is developed into antheridia. Now, if we have to study how the antheridias are present, we have to take the vertical section. When you cut the antheridiophores vertically into slices and observe it under the microscope, the vertical section of antheridia appears to be like this. So now what we are going to study is, we are going to study the structure of antheridiophores. The structure of antheridiophores under the microscope appears to be like this. So as we mentioned, okay, you can see the different parts. They have a long stalk and the disc is present. When you keenly observe the microscope, you can find number of tissues present. The outermost tissue of the antheridiophores on the disc, okay, on the disc is called as the epidermis. What is this epidermis? These are the single layer of cells which is closely arranged. Okay, the epidermis is made up of single layered closely arranged tissues, and they are not continuous. They are discontinuous in nature. Why they are discontinuous in nature? Because they are separated with small air gaps or air pores. Okay, they are separated with small air pores. These air pores are called as chimney pores. They are called as chimney pores. Here you can see small air gaps are present. So these are called as air pores or the chimney pores. This air pores helps in the exchange of gases during the photosynthesis and respiration. Just below the air pores, you can find a large air chamber. Okay, just below the epidermal region, you can find a large air chamber. The air chamber is because they are, okay, filled with the air, and on the basal surface of the air chamber, on the if this is the epidermis, okay, this is the air pore. On the lower surface of the air pore, they are the air chambers, and the basal surface of the air chambers develops number of filamentous structure. This filamentous structure is called as the green in color. These are chlorosynthetic in nature. Okay, they undergo photosynthesis. Okay, so antheridia, when we take the vertical section of the antheridia, outermost layer is the epidermis. Epidermis is single layered and discontinuous because of the presence of the air pores or the chimney pores. Just below the chimney pores or the epidermal region, there are number of air chambers, and on the basal surface of the air chambers is number of photosynthetic filament which helps in the photosynthesis and produces the oxygen and to exchange this oxygen and carbon dioxide, air pores are present. Okay, and I told you it is not straight. The epidermis is single layered and discontinuous, and it is not straight but it is folded inwards. When it gets folded, okay, when it gets folded, they form a deeply sunken region which appears to be flask-like structure. And this deeply sunken region which appears to be flask-like structure, in this flask-like structure they develop a specialized small stalked globular structure called as antheridium. You can find this here. Okay, so epidermis is not straight, it is okay folded inwards to form a deeply sunken region. On the basal surface of the deeply sunken region, they develop a globular stalked structure which is called as antheridium. Since this epidermal region which is folded and in which the antheridium is developed, so this sunken region is now called as antheridial chamber. Okay, so the epidermis is folded inwards, they form deeply sunken region which is called as the antheridial chambers, in which the antheridium is present. How are the antheridium? Antheridium is stalked and globular in nature, and this antheridial chamber opens with a small opening, and this opening is called as ostiole. So this is what is given. VS of antheridiophore shows the epidermis, and with the air pores or the chimney pores, just below the air pores are the air chambers, and inside the air chambers the photosynthetic filaments are present, and this epidermis develops deeply sunken region. In the deeply sunken region, the antheridium is developed. So this deeply sunken region is called as antheridial chamber. Okay, and this antheridial chamber opens with a small opening called as ostiole. And when you see the arrangement of the antheridium in the deeply sunken region or the antheridial chamber, you can see the bigger and mature antheridiums are in the center, where the smaller and immature antheridium or developing antheridium are at the surface. So when you take the section and see, the smaller developing antheridia will be from the outer surface, and in the middle the mature ones will be present. This type of arrangement is called as acropetalous order of arrangement. So the younger immature antheridiums are developed onto the surface and the at the sides, and the mature worms are in the center. This type of arrangement is called as acropetalous arrangement of antheridium. Okay, so next, coming on to the section of antheridium. When you take one antheridium and observe it under the microscope, it appears to be like this. How they are? They are small and globular in nature. This antheridium, when you take the antheridial chamber, if this is the antheridial chamber, the antheridium is present inside the antheridium chamber, and it is attached to the basal surface of the antheridial chamber. The antheridium, this is antheridium. The antheridium is it has two parts, that is the stalk part and the body part. The stalk part is attached to the basal surface. When you consider the stalk is multicellular in nature, and the body part, coming on to the body part is made up of two types of cells: the outermost sterile cell and inner cells. The outermost sterile cell of the body part is called as the jacket cell. The jacket cell's function is to protect the antheridium developing antheridia, and the body cell is made up of antheridial mother cells, or it is called as androcyte mother cells. This androcyte mother cells only will later undergo metamorphosis, that is change in the structures, and they develop the antheridium. You know how the antheridium is developed? The antheridium, where it is developed? The antheridia is developed from the antheridium. How is the antheridium? Antheridium is small globular in structure. Where it is present? It is present inside the antheridial chamber. Okay, how it is? It is divided into two parts, that is the stalk and the body. How is the stalk? The stalk is short and multicellular, and the body is made up of two type of cells. The outermost cell is called as the jacket cell. The inner cell is called as the antheridial mother cell or androcyte mother cell. Okay, now how is the antheridia developed? Antheridia is developed from the antheridial mother cell by the process called as metamorphosis. Okay, so now if you're clear with that, we'll continue with this now. Okay, how do the antheridia develops? You know the structure now. How the antheridia develops? Antheridia develops onto the, okay, in different way, okay, in a stepwise manner. When you take the epidermis, which is sunken deeply inside, okay, the superficial cell, one of the cells of the epidermal layer, okay, they develop into a specialized cell called as antheridial initial cell. Antheridial initial cell will be covered with the other cells also, but this cell will be different from the other cells with its size, structure, etc. This antheridial cell, firstly they undergo first horizontal division, where one cell divides into two. The lower cell is called as the basal cell and the upper cell is called as the outer cell. The basal cell develops into the basal part of the antheridium. The outer cell now develops into the antheridia. Antheridium. How it develops? First, the outer cell now, not the basal cell, but the outer cell. Outer cell undergoes two repeated divisions. When they undergo two repeated divisions, they form four cells. Out of these four cells, the two cells on the upper side develops into the primary antheridial cell and the two cells at the lower side develops into primary stalk cell. The primary stalk cells results into the lower part, that is the stalk region, and the primary antheridial cell results in the formation of body part. The stalk cells undergoes multiple divisions and they form number of multicellular short stalk, whereas the primary antheridial cell undergoes division, that is two anticlinal division and two periclinal division, and they form the 8 cell stage. The 8 cell, in this 8 cell, okay, four cells on the outermost layer develops into the jacket cell, and the 4 cell in the inner regions develops into the androgonial cell. Okay, the jacket cell later divides and they forms the protective jacket or the coat. Okay, and the androgonial cell undergoes repeated cell division and they form androcyte mother cells. Now, the androcyte mother cell will undergo metamorphosis. They change in their structure and shape and they develop the antheridia. Okay, so that is about the development of antheridium. What I have explained there, it is given in the points here. Okay, now how do the antheridial mother cell develop into the antheridia or the sperm? Okay, antheridia is otherwise called as the male gamete or the sperm. So now we will be studying about the spermatogenesis. Okay, spermatogenesis or the antheridia develops by the metamorphosis, that is the antheridial mother cell undergoes changes in the structure and they form the antheridia. How they undergo changes in the structure? They have to become elongated, they have to develop the flagella, okay, and the nucleus has to elongate and they have to become motile. So this is the developmental process. This is the process which is involved. So what is spermatogenesis? It is a process of metamorphosis of androcyte, that is androcyte mother cell, into androzoid. That is androcyte mother cell has to now become motile. So since it is motile, it is called as androzoids, antheridia. Okay, androcytes are developed. So this process is called as spermatogenesis. It occurs in two phases. So how the mother cell develops into antheridia? It occurs in two phases. What is the first phase? It is the development of blepharoplast. Blepharoplast are nothing but the flagellas, and the elongation. Second step is elongation of the nucleus. So first one is blepharoplast. So antheridial mother cell, they become bigger and they develop the blepharoplast, and blepharoplast are granulated structure. Then they become densely granulated and they develop the flagella, which extends outside their body. Okay, and the whole plant body cell also becomes different in structure. The triangular cell, they become triangular in nature. Okay, and they develop the flagella. from the blepharoplast. Next, elongation of androcytes. Androcytes nucleus. Okay, elongation of androcytes nucleus. As the blepharoplast develops and the flagella develops, even the nucleus also elongates. The nucleus also elongates. Okay, as the nucleus elongates, the cell becomes tubular in structure, and because of the development flagella and tubular structure, they become motile in nature. So diagrammatically, it appears like this. So the cell, mother cell, undergoes metamorphical changes. They divide and they form the blepharoplast, the granulated structures, highly granulated, and this blepharoplast develops the flagella, and the nucleus also elongates. As the nucleus elongates, the cell loses its structure, they change in the structure, and the flagella also develops, and now they tend to move. This is how the sperms are developed. Now the mother cell and the side mother cell is now developed into antheridia. Antheridia is now ready to move by developing the flagella. Okay, now it has to find the female gamete. So this is how the male gamete is developed. Okay, the antheridia is developed from the antheridiophores on the antheridial discs. Okay, so in the next session we'll be studying how the female gametes develop, that is the archegonia develops. Okay, so this completes the development of the antheridia. We studied in this session, we studied about the sexual reproduction, we studied about the male gamete producing organ called, that is, and the structure of antheridiophores, and we studied about the VS of the antheridiophores. We studied about the structure of antheridium and development of antheridium and spermatogenesis process. Now the sperm is ready. Okay, in the next section we'll be studying the female, that is female gamete, that is the archegonia. Thank you all.